Hello, everyone, and welcome to today's webinar, Modernize Your Cell Culture, Save Time, Improve Consistency. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speaker for today's webinar, Josh DiGiacomo, PhD, R&D Scientist, Thermo Fisher Scientific. Josh, you may now begin your presentation.
Everyone, my name is Josh DiGiacomo, and I'm a research and development scientist at Thermo Fisher Scientific. I want to welcome you to and thank you for joining our seminar on innovative cell culture. Today, I'm going to be talking about some of our innovative solutions that can hopefully make your cell culture faster, more consistent, and more sustainable. But first, of course, a little background. As I'm sure many of you know, cell culture has been around since the mid-1800s. And while we've advanced a lot since then, many of the classic cell culture products and tools that are still used today in numerous workflows were first developed over 50 years ago, and many have seen very little change in their formulation or application. And again, while these foundational products have led, of course, to many groundbreaking discoveries, updated versions of these cell culture products have features that can save you time, improve the consistency of your results, and can reduce waste in your cell culture workflows and ensure that you're not stuck in the past. Here at Thermo Fisher, we develop new products in R&D by getting input from you as the customers on the workflows you participate in and the biggest issues that you face when executing them. We then use that input to create new solutions which go through a rigorous development process to ensure both quality and that the needs you require are being met. This is actually how we've developed several of the innovative products that I'll be highlighting to you today. I've constructed here a typical generalized cell culture workflow that most of you are probably all too familiar with, no matter what type of research you do. This process, of course, starts with preparing your media and continues through initiating, plating, assessing, and measuring your cells before analyzing your data at the end. Now, this slide is a bit busy, but here is an overview of all of our exciting, innovative cell culture products that can be applied throughout our generalized cell culture workflow. Although I'm happy to talk about as many of these as I possibly can, for time purposes today, we'll be focusing on innovative products that largely apply to the first three steps of the workflow. So first up are our innovative solutions for preparing your complete cell culture medium. First is our advanced media line of products. Most media formulations for traditional cell lines require fetal bovine serum, or FBS supplementation, at some percentage, usually around 10%. While FBS has many components that are very critical for growing cells, FBS is, as the name implies, animal-derived and therefore undefined, so it can be a bit variable from batch to batch. And as many of you surely know, it can also be costly. With our advanced versions of your favorite classical basal media formulations, you can reduce the amount of FBS that you have to add to complete your cell culture medium, sometimes by more than 50% without the need for adaptation or impact to your cell morphology or growth rate for many cell lines. Advanced media is able to do this because we enrich these classical media formulations with components such as ethanolamine, glutathione, insulin, transferrin, and more to make up for the reduced amount of FBS. Advanced media is also available in all of the classical media formulations you see here on the slide. Here's an example of the results when you swap advanced DMEM in for your classical DMEM formulation and reduce the amount of FBS by half of the standard amount. As you can see on the left, there was no change in NIH 3T3 morphology at both high and low confluencies. On the right, you can appreciate that for both NIH 3T3 with 5% FBS and HEC 293 cells with only 2% FBS, that there's no difference in cell culture metrics like count, viability, or doubling time. Now, as for the cost savings, despite the fact that our advanced versions are available for a premium, the reduction in FBS that you're able to achieve can allow for significant cost savings in the long run, as you can see here by calculating on a per bottle basis. So in total, the switch to the advanced version of your favorite classical medium can incur both a cost savings and a reduced dependence on FBS. Next up, for modifications of classical media formulations, is our bench-stable media product line. These are all fantastic products for labs where fridge space is scarce because these formulations have been designed and optimized to remain stable at room temperature prior to supplementation and use. They also come in protective packaging to reduce the risk of light exposure, and personally, I believe this packaging also makes them even easier to stack and store. Bench-stable media can act as a direct substitute for the analogous classical versions after appropriate supplementation with FBS. Here you can see a wide variety of cells cultured in both the original and bench-stable versions of basal media after supplementation. And you can appreciate that the morphologies and doubling times are highly equivalent when cultured in both basal media formulations. Our bench-stable media products are also greener by design. The removal of cold storage requirements prior to supplementation allows for a savings in energy usage. Globally, a switch by scientists to bench-stable media would save 34 gigawatts of energy each year, which is equivalent to the yearly greenhouse gas emissions from more than 5,000 passenger cars. In addition, bench-stable media is contained in our classic Gibco boxy bottle, which is made out of recyclable plastics, and the light protective box provided with the products is made from 100% recyclable material. Moving on to our next product, which is Glutamax supplement. Glutamax is a room temperature friendly alternative to traditional L-glutamine supplementation. For anyone unfamiliar, L-glutamine is a supplement to complete media solutions that is important in nucleotide and amino acid synthesis, as well as glucose metabolism. Typically, L-glutamine is aliquoted and stored frozen because a common issue with traditional L-glutamine is that it degrades into ammonia over time at higher temperatures, which of course can be toxic for cells when it accumulates in culture. Glutamax actually avoids this problem by providing L-glutamine to cells in a much more stable dipeptide form. You can see here over time at 37 degrees Celsius, L-glutamine slowly degrades and produces ammonia over 7 days of storage, whereas Glutamax does not. On the rightmost graph, you can see that this directly translates into the ability to maintain viable cells over time. And for those of you who culture cells with antibiotic supplements as well, we've also recently released a supplement that contains both Penstrep and Glutamax, so you can add 2 supplements with 1 bottle. Glutamax is also a greener by design product for those looking for more sustainable products. Room temperature storage allows for reduced energy consumption to store Glutamax, and without the need to freeze the product, there's no need to aliquot it like you would with traditional L-glutamine, meaning you save on both cost and reduce your plastic waste as well. If you're looking to save even more time by reducing the need for supplementation, it is worth noting that many of our classical media formulations are also available with Glutamax supplement already added. And now we'll look through some innovative solutions for the expansion step of the generalized workflow. First up is our triple express dissociation reagent. Triple is our innovative solution to replacing traditional trypsin. Similar to our other innovative products, triple is able to be stored at room temperature, which reduces the need for aliquoting or warming up your solutions prior to use. In addition to that advantage, triple tends to perform similarly to traditional trypsin while being gentler on surface markers. As you can see here with this data in the leftmost graph from raw 264.7 macrophage culture, dissociation time between triple and trypsin was similar. However, the center and rightmost graphs demonstrate that only with triple dissociation were the cell surface markers CD44 and CD11B maintained for analysis with flow cytometry. In addition, triple does not require the addition of a neutralizer like traditional trypsin does. Here you can see data demonstrating primary neonatal human dermal fibroblasts after 3 passages of culturing utilizing dissociation with either trypsin and trypsin neutralizer or triple express. Across typical cell culture metrics like confluency, cell yield, viability, post-passage growth, and morphology, triple dissociation results in equivalent culture metrics to trypsin and trypsin neutralizer dissociation. This work has been replicated in other primary cells such as endothelial cells and smooth muscle cells, indicating that triple is highly compatible with sensitive primary cell culture and removes the need for adding additional neutralizing reagents. The way that triple is rendered ineffective is actually just through dilution of the solution itself. In this experiment, a 100% confluent monolayer of myoblast cells was treated with triple express at varying dilutions with basal DMEM media for 5 minutes at 37 degrees Celsius. At a 1-to-1 dilution of just basal DMEM with these incubation settings, the triple is rendered ineffective at dissociating the monolayer to the same degree as if no triple was added at all. Triple is, again, a greener by design product through many of the same principles I've been discussing. In addition to saving thaw or warm up time, the ability to store the product at room temperature reduces the need for energy consumption to keep it cold in the fridge and minimizes plastic weight by eliminating the need for aliquots. Now I'll move into discussing some of our exciting new cell culture plastic surfaces for culturing adherent cells. If you've ever used a Thermo Fisher tissue culture vessel before, you've almost certainly used our Nunclon Delta tissue culture surface. This is our standard and most commonly used surface treatment that works for a wide variety of adherent cell lines. However, just recently, we've launched the upgraded version of this surface called the Nunclon Super Cell Culture surface. This new surface was designed to support the adherent culture of cells that can sometimes be more finicky or tricky to grow, such as primary cells, and I'll talk a little bit more about some of the benefits in the next few slides. Here you can see the culture of primary adult and neonatal human dermal fibroblasts when adherently cultured on the delta surface, the super surface, or a competitor premium surface. You can appreciate that the cells cultured for the same amount of time on supra produced a higher cell yield when compared to the other two conditions. This is also reflected in the fluorescent images in the center, where the fibroblasts are much more confluent and numerous on the super surface compared to the delta surface. We also performed a scratch assay with these cells, which is when a confluent monolayer is disrupted by a scratched line and imaged over time to see how fast the cells can refill it. You can see that on the suprasurface, these primary fibroblasts were able to fill the scratch much more quickly than on the delta surface, indicating that there's a boost to both proliferation and possibly cell motility with the suprasurface as well. Demonstrated here is a similar story with primary endothelial cells. The top row is data for human umbilical vein endothelial cells, and the bottom row is data for aortic endothelial cells. You can see once again that the cells cultured on the suprasurface in the same amount of time yield higher cell numbers. In the top row's phase images, we're also demonstrating that endothelial cells retain the ability to form tubes when appropriately cultured on the suprasurface. But perhaps one of our most interesting advantages is in mesenchymal stromal cells. These types of cells are often used in translational development research, and for adherent culture require either an ECM coating or the presence of serum for successful culture. Of course, serum-free media is often preferred, and coating with ECM can be time-consuming and costly. We observe with the suprasurface that these types of cells can be cultured in a serum-free media system with no ECM coating. The left column is a serum-free culture on the delta surface alone. The center is serum-free culture with an ECM coating on the delta surface, and the right is serum-free culture with the supersurface alone. You can appreciate that the cells proliferate and look morphologically similar on supra alone with no need for ECM coating in the sera-free media system as compared to an ECM coating on delta in the same system. We've tested the compatibility of the supersurface with multiple primary cell types and over 25 commonly used cell lines. While not all cell lines or types demonstrate a culture advantage when comparing from delta to supra, we wanted it to be clear that if you do have a cell type that sees benefits on supra, you can easily transition all of your other cultures over to the new surface without a risk of performance loss, so there's no need to keep track of which lines grow on delta and which lines grow on supra. Switching quickly to another interesting type of surface for our cell culture vessels, I'll be talking about our upcell surface. This surface is composed of a covalently immobilized polymer that forms a thin, even coating over the culture plastic and allows cells to grow at 37 degrees Celsius. However, when the surface's temperature reduces below 32 degrees Celsius, the same layer binds water and swells, which results in the release of the adhered cells. This allows for enzyme-free cell dissociation without mechanical disruption, which is crucial for applications such as generating cell sheets or even maintaining expression of surface markers. On the left here, you can observe the recovery ratio of mouse peritoneal macrophages harvested from the upcell surface compared with the recovery ratios of cells harvested either by enzymatic through trypsinization or mechanical through scraping methods. On the right are human bone marrow cells and preadipocytes harvested from the upcell surface through temperature reduction or by trypsinization and then analyzed by flow cytometry for surface marker CD140A. You can see clearly that upcell dissociation helps maintain the marker detection for both of these cell types over the enzymatic dissociation. Lastly, we'll quickly highlight some of the products that can innovate the experimental step of your cell culture workflows. First is our line of Countess automated cell counters specifically focusing on the Countess 3 FL. These handy devices utilize machine learning algorithms to give fast and accurate cell counts and viability measurements. The only work you're required to do is mix a small sample volume with trypan blue stain and add it to a counting slide. Then you just insert and let the Countess do the rest. Here you can see a comparison of count results between manual hemocytometer counting, Countess automated counting, and a cell count from an attuned next flow cytometer across a wide variety of cell types. Across all three metrics for all the cell types tested, the resulting cell counts were highly comparable, which highlights the accuracy of the Countess 3 FL. The time savings associated with using the Countess 3 over manual counting is also highlighted in the table on the bottom. It's worth noting that the Countess 3 is available for use with both disposable or reusable counting slides. For our last highlighted innovative solution, I wanted to highlight our Nunc Edge 96 well plates. These plates were designed to address the issue of the edge effect, in which wells at the edges of multi-well plates can demonstrate higher amounts of evaporation and lead to unwanted variability in your results. The edge plate is designed with a moat that can be filled with liquid to help prevent the edge effect, allowing you to reclaim all 96 well plates for use instead of having to work around the edge or fill the edge wells with non-culture liquid. Here you can see some data demonstrating the percentage of evaporation that occurs in 96 well plate edge wells without a moat. As the graph demonstrates, the Edge 2.0 plate can help reduce evaporation from 10% down to less than 2. On the right, a heat map of variability in cell viability demonstrates the reduction of the edge effect due to the presence of the liquid-filled moat. And lastly, the edge plates themselves are also a greener by design product since they allow you to use all 96 wells instead of having to work around the edge. This results in less plates consumed for the same amount of wells used and ultimately less plastic waste generated over time. I understand we've covered a lot of ground in a short time today, so I also wanted to highlight some of our resources that can help make finding the right product for you even easier. I won't explain all of these today, but I would like to highlight our cell culture select tool, which allows you to easily type in your cell line or type of interest and receive a list of recommended products and information for successful culture. We also have a Gibco Media Select tool, which can help you narrow down and select the media product that's most suited for you by selecting components that you do or don't need, such as phenol red, glutamine, heaps, or more. Lastly, we have both a switch to Nunc and a switch to Gibco tool that allows you to type in the SKU number or product name of similar competitor products to easily find our equivalent product and make comparisons to decide which option is better for you. Quickly, I did also just want to promote an optional survey for anyone who cultures adherent-induced pluripotent or embryonic stem cells. You can scan this QR code or go to the link next to it to take a short 2 to 3 minute survey, which will help us to develop better products to support your iPSC or ESC adherent workflow needs. Any input here is highly appreciated, especially by me since I'm looking to personally lead the development of products to better support these workflows for you. And with that, thank you so much for taking the time to be here and listen today. As a benchtop scientist myself, I have and continue to use a lot of the products I talked about every day in the lab. So I'm happy to field as many questions as time allows.
All right. Thank you very much, Josh, for your very informative presentation. We will now go ahead and move into the live Q&A portion of the webinar. If you happen to have a question that you would like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. Okay, let's go ahead and get started here. The first question that we have says, which of these products do you use in the lab most often?
Oh, geez. Well, uh, a lot of them. But if I had to pick one I use the most, it's probably triple dissociation reagent because I do a lot of adherent cell culture work. So obviously I need a good dissociation reagent to passage those. I also use Supra and Glutamax a lot, but I've probably used all of these at least once over the course of my time in the lab.
All right, wonderful. The next question that we have here says, how does advanced media allow for the reduction of FBS?
Yeah, good question. So advanced media is formulated to contain a lot of components that are actually already naturally present in fetal bovine serum or FBS. And so we add some of those components back into the basal formulation. That way your cells are basically getting the boost of those supplements without needing to add as much FBS to make up for reaching those levels so you can reduce the amount you put in.
All right, fantastic. The next question that we have here says, does the Nunclon supra surface support iPSC growth?
Um, Great question again. I do always get that every time I talk about Supra. Unfortunately, the Supra surface does not support coating-free iPSC growth, but if I can share that one slide again, that was talking about, hopefully that's showing, that there we go. That is showing the QR code here. This actually ties directly into that. So like I said, if anybody is doing adherent ESC or iPSC culture, and the idea of surface-free culture or a coating-free culture appealed to you, I would highly recommend you take the survey to help make sure we can get better products out there for you.
Okay, wonderful. Another question we have here says, do you have any product recommendations for taking care of cells, for example, HDF cells that enter senescence?
That is a great question. Off the top of my head, I know that we have fibroblast specific media. I'm not sure specifically about addressing the problem of senescence, but I can certainly look into that. I don't know if we have a specific product designed for senescence recovery.
All right, we have a couple more here. This one says, can you explain how the upcell surface facilitates cell detachment without enzymes?
Yeah, definitely. Let me see if I can find the slide again. I think a visual is always more helpful here. Let's see. There we go. Yeah, so this special polymer is able to, at a lower temperature, absorb water and swell, but this doesn't happen at higher temperatures like 37, of course, where we're incubating our cells. And so cells can sit on that surface, they can attach, they can proliferate, but once you take them out of the incubator and that temperature slowly drops, that polymer is going to swell, and this results in the release of any cells or anything that was attached to the top of it without enzymatic dissociation.
All right, wonderful. Next question here says, how do Gibco products address sustainability in research?
Yeah, great question. So I hit on it quite a bit throughout several of these products, but I think the through line of all of them is essentially we aim to reduce dependence on cool storage. So a lot of our products are now temperature or room temperature storage or ambiently shipped. So that reduces on the energy need. We also focus on using recyclable materials. So I think all of our boxy bottles are made of highly recyclable plastics. And of course, we're aiming in general to reduce plastic need as well. So a lot of our room temperature storage products that their previous versions required cold storage. You no longer would need to aliquot those to store them cold. You can save on plastic there by reducing your need to aliquot these products since you can store them at room temperature.
Okay, great. Looks like we are getting some additional questions in here, which is fantastic. Reminder to our attendees, if you have not already, go ahead and submit any questions you may have for Josh in the Ask a Question box and click Submit. Okay, this next one says, have you done comparison between Countessa versus uh, V cell counters?
No, off the top of my head, I don't think we have Vicel versus Countess comparisons, but of course in that slide, let's see, there we go. In this slide, you can see we're comparing multiple methods as well and showing that the Countess is able to keep up with other methods such as flow cytometry counting or traditional hemocytometer counting across a wide variety of cell types.
Perfect. Okay. Well, with that, we will go ahead and wrap up here. Thank you again, Josh, for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webinar. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webinar can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. All right, until next time, thank you, everybody. Goodbye.
Thank you.